Semaglutide and tirzepatide are being injected by millions of patients every week, but here is something that doesn't get asked enough. How does anyone actually verify that the molecule in that pen is structurally correct? Amino acid by amino acid, modification by modification. The answer is peptide mapping. And if your team is working on a GLP-1 generic, a biosimilar, or a new analog, this technique is what stands between a regulatory submission and a rejection letter. Hello, I'm Raghuvar Singh Parmar, Director Scientific Affairs at Resolve Mass Laboratories, and we work with pharma and biotech teams every day to characterize peptide drugs at the structural level, sequence confirmation, PTM identification, impurity profiling. Today we are going to walk through exactly how peptide mapping works for GLP-1 peptides, why it's uniquely demanding, and what the regulators actually expect to see. To understand why peptide mapping matters, let's briefly set the stage. GLP-1 analogs have become the dominant class in metabolic disease therapy, type 2 diabetes and obesity, and increasingly cardiovascular outcomes. And as these drugs come off patent, the generic race is accelerating. But unlike a small molecule where structure is relatively straightforward to verify, GLP-1 peptides are large, modified, structurally complex molecules. A single misplaced amino acid, a missing fatty acid chain, an unexpected oxidation, any of those changes can alter the drug's half-life, its receptor binding, and its immunogenicity profile. So structural accuracy isn't an analytical formality, it is the whole game. What is peptide mapping? So what is peptide mapping exactly? At its core, it's a technique that takes your GLP-1 peptide, cleaves it into smaller, predictable fragments using an enzyme, typically trypsin, then separates those fragments by liquid chromatography and identifies each one by tandem mass spectrometry. The result is the structural fingerprint. You are not looking at the intact molecule as a whole, you are restructuring it piece by piece, confirming the primary amino acid sequence, locating any post-translational modification, acylation, oxidation, glycation, detecting sequence-related impurities, and verifying the structure holds consistent across the batches. It's as close to reading the molecular identity card as analytical chemistry gets. The workflow has 5 steps, and each one controls the quality of what comes next. First, sample preparation, purification, desalting, reduction, and alkylation if needed. This removes contaminants that would interfere with the MS signal and ensures efficient digestion downstream. Second, enzymatic digestion. The peptide is cleaved into predictable fragments. The enzyme to substrate ratio, incubation time, and temperature all have to be tightly controlled. Over-digest and you lose fragments. Under-digest and your coverage drops. Third, LC separation. The fragments are chromatographically resolved, which reduces complexity and minimizes the suppression in the mass spectrometer. Fourth, MS-MS analysis. Each fragment is sequenced using high-resolution instruments like Orbitrap or QTOF system. And fifth, data analysis and validation. Expert review on top of software to confirm assignments, identify modifications, and produce a regulatory ready report. Why GLP-1 is demanding? Here is where a GLP-1 peptide separates themselves from routine analytical work. three specific challenges come up again and again. The first is structural complexity. Semaglutide carries a C18 fatty diacid chain on lysine at the position 26 connected through a linker. That modification fundamentally changes how the molecule fragments in the mass spectrometer. The fragmentation patterns becomes less predictable and you need high resolution instrumentation just to confidently assign what you are seeing. The second is stress level impurities. Sequence variants and degradation products often exist well below 1%. You need sensitivity, selectivity, and a method that's been validated to distinguish signal from noise. The third is sample instability. GLP-1 peptides are susceptible to hydrolysis and oxidation during sample handling itself. If your prep protocol isn't tight, you are characterizing an artifact, not the drug. Now, which brings us to what actually differentiates a defensible peptide mapping datasets from one that won't hold up under regulatory review. Four practices matters most. First, use high-resolution mass spectrometry, Orbitrap or Q-top systems with mass accuracy in low ppm range. Anything less leaves you guessing on ambiguous fragments. Second, apply complementary fragmentation techniques. CID and HCD are standard, but ETD adds coverage for heavily modified or large fragments, particularly relevant for semaglutide's acylated domain. Third, optimize your digestion rigorously, not just enzymes type, but ratio, buffer, temperature, and incubation time. And fourth, run replicates. Reproducibility isn't just good practice, it's a validation parameter that agencies check. Now, where it proves its value, let's bring this out of theory. For semaglutide generic development, FDA expects a sameness demonstration, and peptide mapping is the core method used to show that the generics sequence and modification profile are identical to the reference product. For liraglutide stability programs, peptide mapping catches oxidation and hydrolysis degradants that routine HPLC purity methods miss entirely. And for batch release QC, it's the assay that catches a sequence variant before a non-conforming lot ever reaches a patient. At Resolve Mass, our GLP-1 peptide mapping workflows have supported sponsors across all three of those scenarios, from early IND stage characterization through ANDA submission packages. Regulatory expectations. So what do the regulators actually ask for? FDA and EMA require full sequence confirmations by LC-MS, PTM identification and localization, and Health Canada aligned with Q6P peptide for integrity. The short answer for this is all major agencies require peptide mapping for GLP-1 drug approval. This is not an optional study you do to strengthen a submission. It is an expected deliverable. Teams that treat it as an afterthought find themselves generating the data under submission timelines, which is where costly shortcuts happen. So what should you carry forward from today's session? Peptide mapping confirms structure fragment by fragment. It's not a bulk purity assay, it's a structural identity test. GLP-1 peptides require specialized HRMS workflows because of the modification and instability. Regulatory agencies, FDA, EMA, Health Canada require it, and early characterization prevents the late stage surprises that derail submission. and inflate costs. And if you are working on GLP-1 generic or ANDA or biosimilar program and you need a CRO partner who treats structural characterization as a core scientific discipline, not a box checking exercise, Resolve Mass Laboratories supports teams from early stage peptide mapping all the way through regulatory submission. Subscribe to our channel so you don't miss the rest of this series and other informative videos. Drop a comment with which a GLP-1 molecule your team is working on, and thank you so much for watching this session until this. See you in the next one, and until then, goodbye for now.